Welcome to the webinar, The Potential of Psychedelics for the Treatment of Neuropsychiatric Disorders. Hi, my name is Graham Dick. I am the product manager for BioIVT's ADME Product and Services Group. Before I introduce Dr. Tina Mueller, our speaker on today's webinar, I would like to take a few minutes to tell you about BioIVT. Founded in 1981, BioIVT has grown to be the largest provider of biospecimens and research services to the pharmaceutical and diagnostics industry. We are dedicated to providing the biospecimens and services our customers need to help them advance their discovery, preclinical, and drug development programs. We have grown both organically and through strategic acquisitions that have enabled us to develop a comprehensive product portfolio and be the preferred provider for our global biopharmaceutical customers. And because Tina is focused on providing consulting support to design ADME programs, I'll especially highlight the Xenotech acquisition in 2022 that significantly expanded our in vitro ADME research services capabilities. We work with clients to design comprehensive ADME strategies based on their drug development goals and implement studies in our laboratories in Kansas City, the Legacy Xenotech site, and Santa Clara. We have invested in sourcing products globally and in extensive global distribution. We stage products in regions where our customers are located. This helps to ensure short turnaround times and enables us to get products to our customers' labs when they need them. Underpinning all of what we do is our commitment to ethical sourcing, quality, and a sincere desire to work as a partner to the research community. Here we see a snapshot of BioIVT's portfolio, including products used in cell and gene therapy programs, normal and disease state biospecimens, and products and services to evaluate the ADME properties of new drugs. We have developed an extensive infrastructure to collect and produce human and animal-derived cellular starting materials that are used in cell and gene therapy programs. Our ADME group has the industry's largest portfolio of hepatocytes, subcellular fractions, and recombinant enzymes, as well as other products to support ADME tox and DMPK research. Our team of experts routinely works with clients to help them identify hepatocyte lots with the characteristics that meet their specific requirements. Additionally, we help clients design and implement their preclinical ADME research programs and develop the data packages for IND submissions. Our fit-for-purpose study designs include programs in drug metabolism, enzyme induction and inhibition, drug transporter, and other ADME research services. Visit bioivt.com to learn more about our portfolio of products and services. It's my pleasure to introduce my colleague, Dr. Tina Mueller. Dr. Mueller is a scientific advisor for ADME Talk services at bioivt.com. She joined BioIVT in 2023 and consults with customers and internal teams on in vitro ADME programs with a focus on in vitro DDI studies and compliance with regulatory guidance. Tina obtained her PhD in biochemistry and microbiology and earned a master's in environmental sciences from the Swiss Federal Institute of Technology in Zurich. Before joining BioIVT, she was an associate director of research at Cybin, a small biotech company developing new mental health therapeutics based on psychedelics, where she led the company's research program from early through late discovery. Additionally, Dr. Mueller served as a research associate professor at Michigan State University, senior staff scientist at Scripps, Florida, and a professor for biomedical sciences at Kaiser University. Tina, we look forward to hearing your talk on these new applications and clinical development for what is arguably a very old class of drugs.
Yeah, thank you so much, Graham, and welcome everyone to today's webinar about the potential of psychedelics for the treatment of neuropsychiatric disorders. So this is just a quick disclaimer, and with that, let's get started. Here you have an overview of the presentation today. I will start with an introduction into psychedelics, what they are and how they are classified. And then we will move into the more scientific part with the mode of action. So talk a little bit about pharmacology and neuroplasticity. After that, I'll briefly talk about ADME, the little at least that is officially known, and then go into a case study by psilocybin about the development of a next generation example of a psychedelic. I will end with an overview of clinical trials and, of course, a summary and conclusion. So, as you're most likely aware, psychedelics have reached the mainstream news, and here are just a few examples that I found in the news and found interesting. So, in 2018, Michael Pollan published his book, How to Change Your Mind, which was also the basis for a Netflix series. In 2020, Forbes had an article about 5-methoxy-DMT, a psychedelic which actually has been rather popular, as Mike Tyson is a big proponent. Now, fast forwarding a couple of years, the FDA released their first clinical trials with psychedelic drugs in 2023. And just this year, psychedelics have actually been mentioned as potential interests for investors, as this article here in Biospace from March shows. And most recently, in really showing you how psychedelics have been in the public eye, Joshua Siegel and his colleagues, they published a study in Nature about what the brain looks like after taking a psychedelic. Now, these individuals took psilocybin and then had their brains examined and their brains were examined using functional MRI. Now, let that sink in. Imagine taking psilocybin and then being put into an MRI machine. I'm not quite sure how I will feel about that, but the study provided really great insights and was picked up by a lot of news outlets such as NPR or the New York Times. Now, last, the field had a little bit of a setback when the FDA rejected the Lykos application for MDMA assisted therapy in August. So let's now take a step back and first look at what psychedelics are mainly developed for at the moment. And what they do is they target mental health disorders. Now, when we talk about mental illness in this context, we will focus mainly on the following. And these are anxiety disorders, depression, PTSD or post-traumatic stress disorder, and substance abuse disorders, as these are the indications which are mainly targeted with the so-called first generation of psychedelics. Now, as you're probably aware, mental health disorders are a worldwide problem and unfortunately affect millions of people. And just to throw out some numbers here, 1 in 5 adults in the US experience mental illness in any given year, and 1 in 20 actually experiences serious illness. Now, last, I wanted to point out that up to 30% of people with depression do not respond to traditional antidepressant treatments, such as SSRIs. And for PTSD patients, that number is actually even higher and is about 40 to 60%. I do believe that these numbers clearly show that there is unmet medical need for new drugs treating these mental health disorders. Which brings us to psychedelics. So what are psychedelics? Now here is one definition from a review publication by McClure, Bagley, and Roth that I really like. And they define psychedelics as drugs that alter a person's reality, emotion, and cognition without impairing memory or inducing delirium. Now, beyond that, and from a more pharmacological perspective, classic serotonergic psychedelics act by activation of the human serotonin receptor 2A. And what that exactly is, we will learn a little bit more about that later in the presentation. Now, the term itself was coined by Humphrey Osmond, and at that time it was used to refer to compounds that have an LSD-like effect. Now, psychedelic therapeutics are a new class of drugs with a promise to treat mental health disorders. And I want to mention here that potential means that these molecules look promising at the moment. But I want to point out that while there's quite a hype, all correct drug development studies need to be conducted before we know whether that promise can actually be fulfilled. Now, you also probably know that psychedelics have been used in various ritual and religious ceremonies, and of course for recreational purposes for a very long time. Now, it is also interesting to note in this context that psychedelics are generally considered safe at therapeutic doses, and they have a low dependency and addiction potential. And here is just a couple of known psychedelics that you might have heard of. So for instance, psilocybin or LSD, and maybe even the lesser known ones such as mescaline or 2CB. And here's a nice overview of the history of psychedelics, an interesting one to say the least. Now, as mentioned before, psychedelic plants and mushrooms, they actually have been used for thousands of years. And as a result, we have quite a lot of anecdotal evidence and actually know quite a bit about their effectiveness, their side effects, and risks. Now, moving to more recent times, already in the late 1900s, Heffords identified mescaline as the active ingredient in peyote cacti. And then in the first half of the 20th century, Albert Hofmann synthesized LSD and discovered its properties on his famous bike ride on April the 19th. Now, in the mid-20th century, there was really a boom of psychedelics and a wave of early research in the use of these compounds as potential medication for mental health disorders. So like, for instance, in 1957, the magic mushrooms actually were even featured in the Life magazine. Now, this came to an abrupt halt when the Controlled Substance Act was signed in 1970 by Richard Nixon, classifying most of these compounds as Schedule I drugs. Now, in other words, that means that these are molecules that have a high potential for abuse and addiction and no medical use. So another example of a Schedule I drug is heroin. Now, for understandable reason, this brought most legitimate research to a rather abrupt halt. Now, nevertheless, in the 1980s, the target of psychedelics, the 5-HT2A receptor, was actually identified. And since around 2010, there is now a renewed interest in psychedelics, represented here by two milestones. So in 2023, Australia actually legalized psilocybin to be prescribed by a psychiatrist, while on the other hand, just a couple of months ago, as I mentioned, the FDA actually rejected the MDMA-resisted therapy. So now let's switch gears a little and look at the different types of psychedelics. Here we have one way of classifying these drugs, and that is according to their effects on the body. So this is the drug wheel, which I think illustrates the different categories quite nicely. Now, for this talk, 3 of the 7 categories are actually relevant. So we have the so-called true psychedelics, the empathogens, and then last, the dissociatives. I would like to point out that technically empathogens, so for instance MDMA, and dissociatives such as ketamines are technically not true psychedelics. However, most of the mainstream media lumps them in, and for various reasons, I did the same here. So let's now look at these three categories in a little bit more detail. So let's start with the classic psychedelics, which generally produce an altered state of consciousness and are highly psychoactive. So these are the substances that change a person's thoughts, mood, sense of time, as well as the perception of their environment. And here is a list of examples, which we will just look at in a little bit more detail on the next two slides. So let's start with psilocybin. Psilocybin is a substance found in magic mushroom. And you can see the compound or the structure right here. Psilocybin is a so-called tryptamine derivative and is rapidly converted to psilocin, shown here on the right, which has lost the phosphate group. Now, psilocybin has a long history of use in ritualistic and ceremonial contexts. It can be taken orally or smoked, and once ingested, it takes about 30 minutes to feel an effect, and the trip takes about 4 to 6 hours. And those pictures you probably associate with psychedelics, with the geometric patterns and all. Now, that's what very much happens with psilocybin. And last, I do want to point out that the journey or the trip can actually be rather intense. Now, another example is dimethyltryptamine, or DMT, which, as the name implies, is also a tryptamine and closely related in structure to psilocin. It's missing an OH group on the benzene ring. It is interesting that DMT is actually not orally bioavailable, and it has therefore been traditionally used in mixtures that contain a so-called monoamine oxidase inhibitor, such as ayahuasca tea. Now, DMT is fast-acting and results in a much shorter trip. It also produces strong hallucinations and is sometimes called the God molecule, as people often have encounters with God-like creatures, resulting in rather profound experiences. Now, last on this slide, we have 5-methoxy-DMT, which is found in the toad and often smoked. It is an analog of DMT with an additional 5-methoxy group on the benzene ring. And its effect is even shorter than that of DMT, and the trip is most likely one of the most intense. Now, with 5-methoxy-DMT, I also want to mention that it has known safety problems, as one of the metabolites is the so-called bufotenin, which is toxic. Now, moving on to LSD, which has a quite different structure and is a so-called lysergamide and/or an ergoline. This is a synthetic compound and was first synthesized by Albert Hofmann. It's extremely potent with effective doses in the microgram range, not milligram, but microgram range, and also has powerful psychedelic effects. And those last rather long, so up to 12 hours. And LSD has been associated with so-called bad trips. If a person is not in the right mindset, things can go quite wrong. Now, last, we have mescaline here on the right, which is a so-called phenethylamine that is found in cacti. And the compound has a long history of ritualistic use by Native Americans in Mexico. And like LSD, it is very long acting. Now, let's move on to the next class, the so-called empathogens, sometimes also referred to as entactogens. Now, the name implies their somewhat unique characteristic. Taking an empathogen will increase feelings of empathy and pleasure. It makes you feeling connected with the people around you, which is a rather different experience than that of a true psychedelic. Now, at the same time, it relaxes a person and induces a euphoric state and an openness, which can be used to reassess trauma, for instance, during a session. Now, reflecting of the slightly different experience, empathogens have a different pharmacology than classic psychedelics, and they normally do not induce the same strong visual hallucinations we associate with classic psychedelics. Last, on a more negative note, empathogens can cause mood swings, dehydration, something you hear about every so often after rave parties, and depression. And the most typical example is MDMA, so methylene dioxymethamphetamine, sometimes referred to as ecstasy or Molly. And as you probably know, MDMA is most often taken orally and is used quite regularly at parties. Last, let's quickly look at the so-called dissociatives. Again, they are not considered psychedelics in the true sense, but are often included in the discussion. Now, in contrast to the empathogens, dissociatives will make you feel disconnected, so from your own body as well as the environment. And this can, oddly, lead to a calmness and making you being relaxed. It can also provide a relief from pain and will affect how you experience your surrounding. Now, the best known example of a dissociative is ketamine, which is used as a short-acting anesthetic, and esketamine, which is the S enantiomer, with the brand name Spravato, that actually has been approved as a drug for treatment-resistant depression. And one of the advantages is that it has a very quick onset of, for instance, relieving suicide ideation. Now let's transition back to science and start with a brief overview of the current model of the mode of action. First, psychedelics show a complex polypharmacology, and to make things a little bit more challenging, the targeted receptors differ for the different compounds, which I will talk more about in a little bit. What they do have in common is that psychedelics are so-called serotonin or 5-hydroxytryptamine receptor agonists. And looking at the structure of serotonin, shown here on the right, which is the left compound, and psilocin, that is not too surprising, as they are actually quite similar. And the name of psilocin also gives it away, as it is also called 4-hydroxydimethyltryptamine. So most psychedelics are non-selective at the 5-HT receptors, and 5-HT2A agonism is generally regarded as the main indicator of psychedelic effects, while so-called non-hallucinogenic psychedelics do not activate these receptors. Now, in case you are not too familiar with the 5-HT2A receptor, that is a G protein-coupled receptor, or so-called GPCR, and is involved in cognitive function, learning and memory, pain perception, as well as the sleep and waking cycle. So what happens once a psychedelic binds to the 5-HT2A receptor? Now, this activates multiple pathways and initiates a signaling cascade that then in turn leads to downstream signaling effects, such as activation of protein kinases, transporters, and ion channels, all of which are related to the potential therapeutic effects. Now, last, I wanted to mention that psychedelics have been also shown to have so-called neuroplastic effects and induce dendritic spine formation. And now, while most of this presentation focuses, for obvious reasons, on the benefits of psychedelics, there are adverse effects that need to be considered when studying and developing these compounds as potential drugs. So here's a list of possible adverse effects that have been observed. So first, many of the compounds can have a cardiovascular effect with a possible increase of heart rate and blood pressure. Psychedelic administration can also lead to headaches, nausea, and vomiting. Now, these are mostly signs that will subside once they're out of the system. On the other hand, of particular concern here is the next one on the list, namely valvulopathy, which is a damage of the heart valves. This is a side effect that you might be familiar with from other not so great case studies, such as, for instance, the Fen story. Now, furthermore, taking psychedelics poses a risk of causing or inducing psychosis, psychological distress, and there can also be behavioral consequences while and after taking these compounds. With that, let's now go back to the benefits, and as promised, talk a little bit about the pharmacology. And to reiterate, most psychedelics, they do show a complex polypharmacology. And in addition, the different molecules often show different affinities to different receptors. So this slide is giving you an idea of how complex and how different the polypharmacology of these psychedelics indeed are. Now, obviously, I'm not going into the details here, but let me point out a couple of things, more like take-home messages. So we have here the classic psychedelics, so LSD, psilocybin, and DMT. They preferably act on serotonin receptors. Now, but even for those, the 5-HT2A receptor is actually not necessarily the receptor with the highest binding affinity, as these lists here demonstrate. Now, on the right, we can see that ketamine and MDMA have higher activity at receptors other than the serotonin receptors. And of note is the NMDA receptor, which is the N-methyl-D-aspartate receptor, which is a glutamate receptor that plays an important role in learning and memory formation. Now let's take a closer look at just the serotonin receptors. The classic psychedelics are mostly non-selective agonists at all three 5-HT2 receptors. And you can see that here in the table where the binding affinities for LSD, DMT, and psilocin are shown. Now, all of these compounds bind to 2A, 2B, and 2C with similar affinity. And actually, indeed, in the case of psilocin, the affinity at 5-HT2B is higher than that at 2A, which brings me back to an important point about adverse effects. Binding at 2B is linked to valvulopathy, and that needs to be kept in mind when developing these compounds and carefully evaluated. Now, it's also interesting to note that agonism at 5-HT2C can induce anorexia, and indeed, reduced appetite has been reported as a side effect in clinical trials. Now, last, I wanted to point out that these numbers here nicely show that the product psilocybin indeed is not binding to 5-HT2A or to 2C, and whereas psilocin, the actual active drug, does. Let's now look at one of the therapeutic effects, namely neuroplasticity, and why psychedelics are often called psychoplastogens. It has been shown that depression and other mental health disorders can manifest as structural changes in the brain. This means that there are fewer synapses and dendrites on neurons compared to a healthy brain, as shown here in this simple illustration. Now, inducing plasticity and the rewiring of the brain is thought to play a major role in mental health disorder treatments. And indeed, upon a single administration of a psychedelic compound, dendritic spines will form and grow. And also axon branching and synapse formation will start to happen. Now, it is interesting to note that this all happens rather fast in contrast to traditional antidepressants, where the process can take weeks and requires chronic administration. Now, those neuroplastic effects are thought to be mediated by several different transcription factors and pathways that promote neuronal survival, neuroplasticity, and synaptogenesis. Now, one or two of the most critical ones, this would be TrkB and BDNF, which is brain-derived neurotrophic factor. In addition, mTOR also plays an important role in neuroplasticity. It is worth pointing out here that MDMA appears to have a different mode of action, and the effects are mediated via cortisol, prolactin, and oxytocin. Now, in addition to the neuroplastic effects, psychedelics are also thought to have anti-inflammatory and immunomodulatory effects. It's interesting to note that it has recently been shown that a large percentage of depression patients actually have elevated inflammatory biomarkers, and reducing that inflammation might be very beneficial for the treatment of the disorder. So now let's switch to ADME. And just as a heads up, for many of these so-called first-generation psychedelics, ADME was a little bit of an afterthought. One reason for that is the large amount of anecdotal evidence, as well as that these drugs have generally been considered safe. Also, not much has been published, as none of these classic psychedelics actually have been approved. With all that said, moving these compounds from occasional recreational use to a drug candidate, understanding the ADME characteristics is certainly critical. So what I have here are a couple of tidbits. So first here is an example of metabolic stability, which is shown here on the left. In red, you can see the disappearance of psilocin in human liver microsome, which shows less than 30% of a metabolism over about 4 hours. Now, on the right is a proposed metabolic pathway for psilocybin, which was studied in vitro and in vivo. So first, the conversion of psilocybin to psilocin, so the prodrug to the drug, which is catalyzed by phosphatases and esterases. Now, after that, in vivo studies showed that psilocin is mainly metabolized by non-CYP enzymes. So we have psilocin O-glucuronide and 4-HIAA, or 4-hydroxyindole 3-acetic acid, which are the major metabolites found in urine, most likely being converted from or by the action of UGTs and MAO-A, as well as aldehyde dehydrogenase. Now, I need to emphasize here that in vitro metabolism does not necessarily reflect these findings very well. So there's a slight discrepancy between in vitro and in vivo metabolism. Moving on to inhibition, most tryptamine analogs like psilocin inhibit CYP2D6, albeit with a fairly high IC50. Now, some of them also act on CYP1A2 and have weak inhibition at 2C8 and 2A6. Not much has been published with regard to transporters. Generally speaking, psychedelics are expected to be substrates and/or inhibitors for the monoamine transporters. So serotonin, dopamine, and norepinephrine transporters, SERT, DAT, and NET. Now, these transporters participate in the regulation of the monoaminergic pathways, of which a disturbance could lead to psychiatric disorders and neurodegenerative diseases. So here are some binding affinities to the 3 transporters, which are all in the low micromolar range for DMT, and psilocin for NET and SERT. Now, one publication determined actually IC50s for the OCTs, of which OCT1 primarily acts in the liver, OCT2 plays a major role in the kidney, and all of them are expressed in the brain as well. And especially OCT1 is known to be affected by many drugs, such as, for instance, metformin. And you can see that MDMA actually has surprisingly low IC50 for OCT1 and 2. However, psilocin does not appear to inhibit any of the OCT transporters. With that, let me switch gears and present to you the early stages of the development of a novel psychedelic therapeutic. This is an interesting new area because while these first generation of psychedelic drugs show promise in their clinical trials, there's clearly a need for so-called second generation psychedelic therapeutics. Now, these are based on new chemical entities and can be designed to have improved properties, such as, for instance, oral bioavailability or a shorter duration of action. So now let's look at the case study of a so-called phenethylamine. Now, phenethylamines have a general structure shown here on the left, and 2,4-CB is a well-known representative. It has some good characteristics for further development, such as a high selectivity for 5-HT2A over 2B and low potencies at that. It also tends to produce fewer anxious ego dissolution than, for instance, psilocybin. On the other hand, they are often long-acting. So in this program, a compound called CYB210010, or short 2110, was developed. Now, 2110 has a 2CX structure like 2CB, so it is a 2,4,5 substituted phenethylamine. And here are some results from in vitro efficacy studies testing the binding, actually the efficacy at the 5-HT2 receptors. The data showed that 2110 indeed has very high affinity at the 5-HT2 receptors, and of note, it has a high selectivity of 2A over 2B. Now here on the bottom, there is data showing the readout of the so-called head twitch response assay, which is a behavioral readout in mice that measures exactly what the name implies. The mice are rotating their heads from side to side. And simply said, that rotating of their head, that twitching of their head, that is correlated to the binding at the 5-HT2A receptor. And two things can be learned from these assays. So the intensity, in other words, how many times the animal actually shakes their head, is correlated to the dose, and the duration of the effect can be compared to known standards. And here's a graph comparing the different doses to DOI, which is shown here in red, which is one of the longest acting psychedelics known. Now in light blue is the highest dose of 21, showing a more intense and a longer response, but still having an effect after 6 hours. Moving on to in vitro metabolism, 2110 had a medium half-life of about 55 minutes in rat liver microsomes and was stable in human, mouse, and dog. The same species differences emerged in hepatocytes, where the compound was stable actually over 24 hours in humans and slowly metabolized in rats. Now, overall, this indicates that 2110 is a poor substrate for phase 1 and 2 metabolism. Keeping in mind that metabolism was slow, metabolite profiling was done in rat hepatocytes and showed monooxygenation leading to sulfoxide formation, as well as the sulfoxide formation with N-acetylation and O-glucuronidations. Now, last and not too surprising, no metabolites were detected in human hepatocytes. And early in vivo experiments showed some interesting findings as well. Here's an example of a PK study in rats and a comparison of IV and oral administration, which are shown on the left and right panel respectively. In light blue is the concentration in plasma, and in dark blue in the brain over time. Now, comparing the IV and the oral administration, it can be seen that 2110 is orally bioavailable, and it's just about 20%. It's also noteworthy that the molecule shows a very rapid distribution into the brain, seeing a rapid increase in the brain concentration, as you can see in the graph, and indeed had a rather high brain to plasma ratio that will need further evaluation. So in conclusion, 2110 showed a high potency and functional selectivity for the 5-HT2A receptor and robust and durable in vivo efficacy. However, it probably needs a little bit further fine-tuning, and for instance, introducing soft spots may enable a shortening of the duration for instance. So now let's move on and talk about clinical trials. Here is an overview of the clinical trials on clinicaltrials.gov at the end of August. So it's a couple of months. However, what I found at that point was that there are quite a few completed trials. There are over 40 for psilocybin and over 50 for MDMA. LSD is trailing a little bit with just 15. There are also many ongoing trials with over 100, for instance, for psilocybin. Now, the main indications are phase 1 with healthy volunteers, and the mental disorders I mentioned at the very beginning are the main indications. So, substance abuse, PTSD, depression, as well as some, of course, additional indications as well. Now, I also want to point out a couple of phase 3 trials, and there are some which one of them has been completed and they are starting or are in progress. Lykos had its MDMA assisted therapy for PTSD, and then ketamine was tested for suicidal depression by Janssen, and 4 trials are looking at psilocybin for either treatment-resistant depression, TRD, or major depressive disorders, MDD, which is done by Simon Uzona and the University of North Carolina. Now let's briefly look at the phase 3 trial with MDMA, which here we have a publication by Jennifer Mitchell from the University of California in San Francisco and colleagues, and of course members from MAPS, the nonprofit organization associated with Lykos, and its founder Rick Doblin. Now this study was published in Nature Medicine in May of 2021. Now the drug was, the title as the title says, MDMA, and the indication was PTSD. As mentioned, it was a phase 3 trial with randomized, double-blind, placebo-controlled testing of efficacy and safety. I would like to point out here that patients with comorbidities were permitted in the trial, but psychiatric medications was stopped before entering the trial, so there was a washout period. The results showed improvement of the PTSD symptoms compared to the placebo, and no adverse effects were observed. With that, the authors concluded that the treatment was effective, safe, and well tolerated. Now, here are 2 publications about phase 2 trials with psilocybin, both by Guy Goodwin from Compass Pathways. In the study on the left, with collaborators from the King's College in London, as well as others, we have a single dose psilocybin for treatment-resistant depression. It included 3 different doses and was a randomized, double-blinded study, and it tested for efficacy and safety. Now, it also showed, or the results showed, that psilocybin was effective at reducing the depression at the highest dose, namely 25 milligrams. Now, the study on the right is noteworthy because it allowed participants to stay on antidepressant drugs. In short, it showed that psilocybin was effective actually when co-administered with an SSRI. Now, with all that said, both of these trials had low enrollment. The one on the left, the NEJM study, had about 90 participants, and the co-treatment, the one on the right, just 19. So in other words, there's clearly a need for larger trials at this point. So with that, we did talk a lot about the promise of these drug candidates, but I wanted to mention once more that challenges and concerns should not be ignored. The risk of adverse effects exists, and this also applies to bad trips, as if once a person has a bad trip, not much can be done beyond keeping them safe at that point. We should also not diminish the trip itself, as it can be a transformative experience. So preparation with the help of psychotherapists is critical. We also need to be aware that while there is a lot already known and the field has made great progress, much of the knowledge is anecdotal and does not adhere to current scientific standards. And with that said, additional research needs to be conducted. We need to better understand receptor binding, signaling pathways, as well as the mode of action of the different compounds. And then there's, of course, the issue of designing the correct trials. This starts with addressing the placebo effect and how to best demonstrate efficacy and safety. So, for instance, the correct patient population needs to be enrolled in a clinical trial. And the question of expectancy bias needs to be addressed, as well as the functional unblinding happening in most studies. With this, we are at the end. So let me quickly summarize this presentation. Psychedelics are developed as new mental health treatments for psychiatric disorders, such as, for instance, depression, anxiety, as well as post-traumatic stress disorders. We have talked about the three main classes in this context. We have the classic psychedelics, such as psilocybin or DMT. And then there are often empathogens like MDMA, and then the dissociative anesthetics, such as ketamine, they're often included in the discussions. Now, classic psychedelics act as 5-HT2A receptor agonists and induce neuroplasticity and anti-inflammatory pathways. And last, in vitro and in vivo preclinical studies, as well as clinical trials, they show promising data for efficacy as well as safety. Now, beyond the first and second generation of psychedelics, and not discussed here, I did not cover two specific topics that clearly also warrant a discussion, but they just went beyond the topic of this presentation today. But that will be the non-hallucinogenic psychedelics, and then, of course, microdosing. And with that, I will finish and thank you all for your attention. Of course, I will take any questions.
Thank you, Tina. And as we wait for questions, I just want to mention that you can receive notifications for future webinars and other resources